Organs of special senses. Organ of taste, gustatory organ, and the ear, organ of hearing and equilibrium. Main questions of the lecture. Organ of taste, it's not the tongue. It's represented by taste buds of the tongue. There are about 2,000 of them. Taste buds located in the papillae epithelium of tongue. There are 3 types of papillae containing taste buds: foliate papilla, circumvallate papilla, and fungiform. And in the epithelium they contain the taste buds. They occupy the whole thickness of the epithelium covering the papilla. Taste buds and foliate papilla of the tongue. They located in the epithelium covering the papilla, so inside parts of them. Taste bud under electron microscope and following silver impregnation of section, where the dendrites of sensory neurons visible. This is a taste bud under very high magnification of light microscope. This is the epithelium, stratified, squamous, non-keratinized epithelium. And this is taste bud, these cells and taste pore, the entrance to taste pit. There are 3 types of cells: receptor cells, sense epithelial microvilli contain chemoreceptors, which connected specifically with these chemicals of food or drinks which enter the oral cavity. After specific interaction, the conformation changes of receptor complex take place, the ion canals open, and excitation of receptor cells take place. And this excitation accepted by dendrites of sensory neurons, which convey the information into the brain. Supporting cells between basal membrane located the small basal cells, stem cambial cells which proliferate, and the daughter young cells differentiate and replace the older aged, defected and died receptors and supporting cells. The ear, organ of hearing and equilibrium, much more complicated in structure and functions. It consists of external ear, light brown, middle ear, staining in pink, and internal ear, staining blue. External ear consists of auricle, this elastic cartilage inside, covered by skin. Then external acoustic meatus, like a tube. Its wall in first third consists of elastic cartilage and later it consists of temporal bone. And third part is tympanic membrane, consists of two layers of dense irregular connective tissues, lining from outside by epidermis of skin, and from inside simple cuboidal epithelium. Middle ear consists of tympanic cavity filled in by air and connected with nasopharynx by auditory Eustachian tube. As a result, the pressure inside tympanic cavity and outside in environment became the same. There are three auditory ossicles, the first connected with tympanic membrane, the last close the oval window, the entrance into internal ear, into vestibular scale of cochlea. And there is also vestibular cochlear nerve demonstrated in the picture, which leave information from internal ear. So the internal ear consists of cochlea, vestibulum, and three semicircular ducts or canals. This slide demonstrates auditory ossicles, malleus first, incus intermediate, and stapes, which close the oval window. Internal ear, the most complicated part of the ear. It consists of bony and membranous labyrinth. Bony labyrinth is a system of cavities and canals in temporal bone. It consists of cochlea, vestibulum, and three semicircular canals, ducts. These extensions, ampulles. This is the oval and round window of vestibulum. This is a membranous labyrinth, consists of dense regular connective tissue. Membranous labyrinth located inside bony labyrinth, so its shape quite similar to bony labyrinth. It consists of membranous cochlea. In vestibulum, triculus and sacculus, and this triculus, three membranous semicircular ducts connected. And this picture demonstrates the location of receptor cells in corti organ, in cochlea, and two macules, and utricle and sacula, and three Cristae ampullaris in three ampulles of three semicircular ducts. It's axial section, showed by this red line here, here, and here. Cochlea consists of canals, make 2.5 cycles around modulus, bony core of cochlea. So it's a section. You see this cochlea core, modulus, and 5 sections of canal of cochlea. So this is a bony labyrinth, and inside membranous labyrinth, triangle, membranous labyrinth inside. And there is also spiral crest, like extension of modulus, and inside spiral ganglia located. The axons compose the cochlear nerve, which bring out the information from Corti organ. So in every section of Bony canal you see the triangle membranous labyrinth, which dividing the Bony labyrinth for two scales, Scala vestibuli and tympani, so Scala vestibuli and tympani, and membranous labyrinth, sometimes called Scala media. This is a cross-section of cochlea under low magnification of light microscope. This is a Bony labyrinth, modulus, spiral crest, membranous labyrinth, triangle, vestibular membrane and Scala vestibuli, tympanic membrane and Scala tympani. Membranous labyrinth, sometimes called cochlear duct. In spiral crest and modulus you see the spiral ganglion, and this is organ of Corti, spiral organ. Let's see the square under middle magnification. This is a Bony labyrinth, Scala vestibuli and tympani, vestibular membrane and tympanic or basilar membrane, Corti organ, and spiral ligament, thickening of Bony labyrinth periosteum, as well as lymph, also the thickening of periosteum of spiral crest. And this is Corti organ and inner tunnel of Corti. Let's see the square at high magnification. This is the borders of membranous labyrinth or cochlear duct or Scala media, vestibule membrane, connective tissue membrane covered by simple squamous epithelium, endothelium both sides. It's divided the Scala vestibuli. And basilar membrane, also dense connective tissue. One side contains a simple squamous epithelium, another side contains the organ of Corti. Cochlear duct filled by endolymph and Scala tympani and Scala vestibuli filled by Perilymph. This is a spiral ligament covered by stria vascularis or vascular strip. Consists of pseudostratified epithelium containing the blood vessels. It's only one exception in the body. Usually epithelial never contain blood vessels. But this unique epithelium secrete endolymph which fill the cochlear duct. This is lymph, vestibular lip, upper lip. This tectural membrane, gelatinous-like structure with collagen fibers which cover organ of Corti. In the center of organ of Corti located the inner tunnel, close to the center, close to modiolus, located the inner hair and supporting cells, and close to periphery to spiral ligament located the outer hair and supporting cells. This figure demonstrates organ of Corti, the schematic drawing, and this is very high magnification of light microscope. This is a basilar or tympanic membrane, Scala tympani, here. This is a cuboidal or squamous epithelium lining the Scala tympani. This is a basilar membrane. This Corti organ. This is the lymph. This pectoral membrane here. This is a spiral crest. These bipolar sensory neurons. This is the inner tunnel composed by S-shaped pillow cells. Inside the tunnel, dendrites of sensory neurons are going. They reach the receptor hair cells. They are basal parts, and they accept information from receptor cells and convey them through the axons into the brain. This is inner tunnel, and there is one row of receptor sensoepithelial cells and one row of supporting cells. So internal receptor cells, internal supporting cells, and 3 to 5 rows of outer receptor or hair cells. 3 dark points are the nuclei of receptor cells, and they located at the supporting cells, also 3 to 5 rows of supporting cells. You see 3 nuclei of them. This is pectoral membrane, and here's Stereocilia of receptor cells connected this pectoral membrane. If you look the surface organ of Corti using the scan electron microscope, you can see the Stereocilia of outer hair or auditory receptor cells, and inner, one row of inner receptor cells, Stereocilia, in one line. This is the outer hair and supporting phalangeal cells under scan electron microscope, and hair, receptor and supporting cell, the schematic drawing. Receptor cell, Stereocilia here, supporting cells, has long process called phalangeal process, is the reason why the supporting cells called phalangeal cell. And these processes make like umbrella the apical cuticular plate, and the plates of all supporting cells connected and separate the Stereocilia. So these are receptor cells, these are phalanx processes of supporting cells. This is a schematic drawing of inner hair cell and outer hair cells. Both of them have Stereocilia with mechanoreceptors, the nuclei, cytoplasm, with basal part located, connected the dendrites of sensory neurons of spiral ganglia. So you acquainted yourself with the structure of the ear from organ till cellular level. You see external, middle and internal ear, the structure of cochlea, acoustic nerve. This is one section, and corti organ, pillar cells and tunnel and tectoral membrane. This is inner and outer hair, receptor and supporting cells. These are dendrites of sensory neurons. How the ear working, functioning? So histophysiology, the mechanism of hearing. Once somebody speaks, this vibration of air, acoustic waves or sound waves, accumulated by auricle, concentrated in external acoustic meatus, and these waves make the vibration of movement of tympanic membranes and ossicles, which increase and convey the vibration to Perilymph of Scala Vestibuli at the apex of cochlea through the connection helicotrema. This vibration movement conveyed to the movement of Perilymph of Scala tympani, and it's induced the vibration of tympanic membrane or basilar membrane and cortive organ. Cortive organ vibrate, is moving, and Stereocilia of hair cells touching, irritated by tectoral membranes, and conformation changes in mechanoreceptors take place, ion channels open, and excitation of hair cells take place, and this excitation accepted by dendrites of sensory neurons of spiral ganglia, and on the axons conveyed to the brain, first into the intermediate part in brain stem, and then conveyed to the brain cortex, auditory cortex, where this information from receptor cells analyzed and images of sound appeared. Organ of equilibrium detected the position and movement of the head. It consists of vestibulum with semicircular ducts, consists of bony and membranous labyrinth, and location of the receptor cells demonstrated by yellow color. Two macules, macula of sacule and utricle, and three Cristae ampullaris in ampulls of semicircular ducts. This slide demonstrates the section of macule of utricle and Cristae ampullaris in ampull of one of semicircular duct. In general, this is a bony labyrinth. It's bone, filled in by Perilymph, and inside membranous labyrinth, filled in by endolymph, and this is macula, and Cristae ampullaris here. This is a macula. Macula detects the linear acceleration and gravitation, as well as vibration in macula of sacculus. There are two types of epithelial cells in macula. Receptor cells containing Stereocilia with mechanoreceptors, about 100 Stereocilia and 1 kinocilia, which can move. Supporting cells between dendrites of sensory neurons of vestibular ganglion. Otolite membrane, a gel-like substance containing the otolites or otoconia, crystal of calcium carbonates. When the linear acceleration or deceleration take place, or changing in gravitation or vibration, the endolymph of membranous labyrinth, utricles and sacculus is moving, vibrate, and it induces the vibration movement of otolite membranes and crystals, otolites, irritates the mechanoreceptors of Stereocilia of heron receptor cells. They exciting, and this excitation accepted by dendrites of sensory neurons and conveyed to their bodies, pericardions, in vestibular ganglia, and then information on the axons conveyed to the brain stem and central part of the vestibular analyzer. This is the hair cells, the Stereocilia and 1 kinocilia, nucleus, and dendrites of sensory neurons branching around the cell's basis. This is Stereocilia and kinocilia under scan electron microscope, and at the transmission electron microscope you see the transverse sections of Stereocilia and kinocilia, which contain 9 pairs of microtubules and 1 pair of central microtubules. So it can move. Cristae ampullaris, located in the ampulls of semicircular ducts. They detect angle acceleration and deceleration. Semicircular ducts filled in by endolymph, and in the place when they connected to vestibulum, they have extension which contain Cristae ampullaris. And the base is extension of the thicker part of membranous labyrinth. These gelatinous cupula around. The size about 1 millimeter. And two types of epithelial cells located there, like in macula. A receptor, hair cells, sense epithelial cells. The Stereocilia contain the mechanoreceptors. And between located supporting cells and dendrites of sensory neurons of vestibular ganglia branching around basal parts of receptor cells. How it's work? When the head move in one plane, the corresponding semicircular duct erect, located in the same plane. The endoleaf inside move and induce the movement of cupula and Stereocilia embedded mechanoreceptors activated and receptor cells exciting, and this excitation accepted by dendrites of sensory neurons of spinal ganglia and the axons convey information to the brain for analysis. And this information analyzed and synthesized, and the image concerning the location of the head and body in the space appeared. We live in 3 dimensions space, so there are 3 semicircular ducts, 3 ampulles, and 3 cristae located in 3 different planes. This is a ear development schematic drawing. The ear development is quite complicated process and in embryogenesis. And there are several sources for different parts of ear development.